Good morning. Thank you all for being here. My name is Ruth Camel, and I'm from Princeton University. I'm going to be talking to you today about Princeton's iGEM project. We're a team made up of undergraduates, and we're going to be presenting some of the work that we did this past summer. Excuse me. Our project is entitled Program Differentiation of Mouse Embryonic Stem Cells Using Artificial Signaling Pathways. So our overall research objective is to apply synthetic biology in stem cell research. It has been shown before how to take embryonic stem cells, proliferate them, and direct them into differentiating into a variety of cell types, such as muscle cells, neuronal cells, and insulin-producing beta cells. However, what is not known is how to take embryonic stem cells and differentiate them into three-dimensional patterns of tissue. So our approach to program tissue generation is to construct artificial cell-cell communication circuits and embed them into embryonic stem cells. These cells then have the capacity to perform signal processing on the communication signals that they receive and send. One of the effects of this signal processing is the ability to make decisions, and one of the more important decisions that a cell makes is whether to differentiate into a particular cell type or not. And that's where directed differentiation comes in. We also use precise cell placement, which provides initial cues to the tissue to be differentiated in 3 dimensions, so that we can essentially create an artificial morphogen gradient. So now I'm going to give you a quick overview of Princeton's iGEM projects. The first system that we designed genetically engineers sender cells to allow them to communicate with receiver cells and direct them into differentiating into 1 or 2 different cell fates. What this does is gives us a basic ability which we can then use to create more sophisticated systems that are able to do programmed tissue engineering and essentially give us the ability to create tissue by design. Now, this system could have applications in things such as regenerative medicine or injury repair of tissues. The second system that we designed also uses cell-cell communication, but here the receiver cells are programmed so they are able to remember the instructions given to them by their neighboring sender cells, and hence they're able to hold state. The third system that we designed allows cells to do population density detection and accordingly differentiate into insulin-producing beta cells. So what this means is that the embryonic stem cells are able to detect if they've reached a critical density or not, and then accordingly auto-regulate themselves to differentiate into insulin-producing beta cells. And this system has applications in treatment of type 1 and type 2 diabetes. So a little bit about embryonic stem cells. Embryonic stem cells are particularly cool to do genetic engineering in because they have the potential of differentiating into each one of the 3 germ layers, which are endoderm, mesoderm, and ectoderm. And then from there on, they can differentiate into every single one of the cell types present in the body, such as bone and muscle. So depicted here is a circuit, a very well-studied differentiation pathway that shows how differentiation occurs up to mesoderm and endoderm. As you can see, the circuit, though well studied, is quite complex. But what we can take away from this is that sulfate regulators drive the differentiation process. So we can use this fact to our advantage by constructing artificial circuits that control these master sulfate regulators and turn them on in appropriate dosages and combinations. Depicted here is a pathway that involves this sulfate regulator. Involves the sulfate regulator shown here, which is MyoD. This pathway here describes how embryonic stem cells get taken into muscle precursor cells. MyoD, as you can see in its natural environment, is both inhibited and activated by a variety of endogenous signals. So our approach is to construct an artificial signaling circuit that overrides these endogenous signals and allows us to control MyoD expression regardless of what endogenous signals might be turning it off or turning it on. So to construct these artificial circuits, we use stem cell biobricks. Stem cell biobricks are made up of 4 main parts. We have a promoter, we have a gene of interest, we have something called the iris, which is an internal ribosome entry site, which allows us to make bicistronic constructs, and we have a marker, which could be a fluorescence marker or an antibiotic resistance marker. This construct is then inserted into a lentiviral backbone, which is used so that we can stably insert the biobrick into the mammalian genome. To create a library of stem cell biobricks, we use these 3 restriction enzymes shown here so that we are able to swap the promoters and genes as desired and dictated by the circuits that we're building. A stem cell biobrick registry is under construction and will be available shortly. It will be open source and open access and will be linked to MIT's registry. Each of the biobricks in this registry will be optimized for mammalian cells. Currently, we have 120 biobricks, of which 70 are functional, which means that they have been verified in embryonic stem cells. Most of these biobricks have been constructed by Sairam in the Weiss lab, and some of them have been constructed by Sil in the Weiss lab and Christoph in the Lemishka lab. So the biobrick delivery mechanism that we use are lentiviruses, because otherwise it's very hard to genetically engineer or manipulate embryonic stem cells. So the first step in this process is to create a bacterial plasmid, that is a plasmid in bacteria. We then take this plasmid and transfect it into mammalian cells that have the potential of creating virus. We can then harvest this virus and use it to infect embryonic stem cells. So if we had to create a circuit that had more than one basic biobrick, we could just take two lentiviruses or more with different kinds of biobricks in them and then use them to co-infect the embryonic stem cell, thereby delivering the circuit into the cell. So to reiterate, for multi-element circuits that we wish to build, we simply co-infect with multiple basic biobricks. And when we want to build different biobricks, we simply swap the elements in and out as desired instead of the concatenation that's required in bacterial biobricks. Depicted here is the first circuit that we built this summer. And I'm going to talk a little bit about the simple induction circuit that uses this biobrick. RTTA is a gene that's constitutively produced. In the presence of exogenous DOX, RTTA and DOX form a dimer that is able to activate the TRE promoter, which drives expression of both red and green fluorescence. So here are some of the results we saw. These are the first results that our team saw with a stem cell biobrick in embryonic stem cells, so we are very excited about that. As you can see here, when we don't have DOX in the system, we have no fluorescence, and with DOX present in the system, we see both red and green fluorescence. Here are some of the results with the first differentiation biobrick we used. PAC6, shown here, is a master sulfate regulator that is implicated in neuronal differentiation. This biobrick has been infected in HeLa cells, and we can see on the right-hand side that we have some dendritic morphology being seen in HeLa cells, which are generally spherical. So this dendritic morphology is reminiscent of neurons. We are currently investigating the effect of PAC6 in embryonic stem cells. Here's a list of the biobricks that we contributed to the stem cell registry this summer. We have 6 working lentiviruses, which means we've designed them, constructed them, and tested them in embryonic stem cells. We also have a few plasmids that are under verification and a whole bunch that are still under construction. Now I'm going to turn you over to my colleague Safi, who's going to talk about cell-cell communication.
Thanks, Ritu. So as Ritu mentioned, the first component in our plan to execute pattern tissue generation is to perform some sort of artificial cell-cell communication between mammalian cells. In order to do this, Sairam Subramanyam in our lab adapted the Lux bacterial communication system to mammalian systems. And this is a totally non-trivial transition, as we'll see while I'm explaining the senders and receivers. The sender cells contain the 3 biobricks that you see here. LuxI, ACP, and AAS are all constitutively expressed. AAS activates ACP, which is a critical component of the fatty acid synthesis, or FAS cycle, and an intermediate of that cycle is used by LuxI to catalyze AHL production. So one optimization you'll notice here from mammalian cells is that LuxI and ACP have been codon optimized from the bacterial system. Once the senders send out the AHL, it'll freely diffuse into the surrounding media and can be detected by receivers in the following way. LuxR is being constitutively expressed in the receiver cells, and GFP, or you can imagine any other gene of interest, for example, master sulfate regulators, can be controlled under the PLux promoter. In this case, you'll notice that the PLux promoter, or the LuxR protein, is a mammalian-optimized version of the bacterial LuxR protein, in addition to 3 other components. Furthermore, the PLux promoter contains 7 Lux operator regions fused to a mammalian min-CMV promoter. So when the receivers detect AHL, it'll simply bind to the LuxR which is constitutively expressed, and that dimer can bind to the PLUX promoter and drive GFP expression or expression of any other sulfate regulator. This is a verification of the mammalian senders, and we can verify them using bacterial receivers because we know the bacterial construct works properly. In the negative control case you see in the left chart, we notice that the sender cells, which are missing one of the components required to produce AHL, do not send out AHL, and correspondingly the bacterial receivers show very little fluorescence, as we would expect. To the contrary, when the mammalian senders can produce AHL, the bacterial receivers respond by producing large amounts of GFP. We can also verify the mammalian receivers in a slightly different manner by simply introducing our signal AHL exogenously. We see here that we have constitutive LuxR production and DS-red production, which indicates that LuxR is being produced. And we have GFP under the control of the PLUX promoter. So when we introduce AHL into the system, we expect that it'll bind LuxR and drive GFP production. Indeed, this is what we see experimentally. In the absence of AHL, we only see constitutive DS-red production. And when we introduce 0.5 millimolars of AHL exogenously, we see that not only do the stem cells express DS-red, but they also glow green. The first of three systems which we design and implement to utilize this artificial cell-cell communication is a multiplexer for cell fate determination. Here on the left, we see a genetic circuit corresponding to that multiplexer, a truth table, and a corresponding logic gate diagram. You'll see in the truth table that we have AHL, which is our primary input here, which is being sent from receiver cells, sender cells, and ATC, which is basically an on-off switch for the system. We'll see how that works in a second. This multiplexer is important because it allows us to perform spatial patterning into multiple cell types, because you can imagine placing sender cells in a certain configuration, which will produce an AHL gradient in a certain shape or pattern, and that'll correspond to the pattern of differentiation which the receivers will take on. In the first case, where there's no input to the system, we see that we get no MyoD or NGN1 expression. These outputs are master sulfate regulators. MyoD is a master sulfate regulator for muscle precursor, and NGN1 is a master sulfate regulator for neuronal precursor. Since we don't have AHL in the system, LuxR cannot drive MyoD or LAC-I expression. And since we don't have any ATC in the system, TED-R is free to repress NGN1. So the system is effectively off, and this allows us to proliferate the stem cells until we want to turn the system on. When we've turned on the system with ATC, and when these receiver cells are near AHL sender cells, we see that we only get MyoD expression because AHL binds LuxR and drives MyoD expression as well as LAC-I, and LAC-I is a repressor of NGN1. So even though TED-R is deactivated by the ATC, LAC-I effectively represses NGN1, and we don't get any of that. We only get MyoD expression. In the case where these receiver cells are too far from an AHL gradient to be affected by it, we see that only ATC affects the system, binds TED-R, and de-represses NGN1. So we get NGN1 expression. However, no MyoD is in the system because AHL is not there to bind LuxR and drive MyoD expression. So this is an example of a biobrick that was constructed in our lab, TRE, MyoD, IRC, GFP. Basically, these embryonic stem cells respond to exogenous doxycycline, which binds the TRE promoter and results in transcription of MyoD, which, as you remember, is a master sulfate regulator for muscle precursor. So when we introduce doxycycline into the system, we see that these stem cells not only glow green, but you also see some morphological change, resemblant of myoblast or muscle precursor cell. So you'll see on the lower left corner that a lot of cells start clustering together into morphology that shows that they're differentiating into myoblast. And right now we're testing the system basically under the Lux system. So instead of the TRE promoter, it'll be driven by the PLUX promoter, so that instead of responding to exogenous doxycycline, these embryonic stem cells can respond to nearby AHL sender cells and differentiate into myoblast according to that signal. To simulate this behavior, we implement a spatiotemporal stochastic model of diffusion and biochemical interactions in this multiplexer. It was adapted from Drew Endy's stochasterator, and basically we wrote an interface in MATLAB that allows us to place cells in a grid and see what sort of dynamic patterns emerge. So you can imagine this is useful not only to understand the basic behavior of the system, but we can see that ultimately when we want to do tissue by design, we can place cells, sender cells, in non-trivial configurations and see what type of differentiation patterns emerge. So here we'll see that we've placed sender cells in 3 small clusters in a triangular shape. They'll form an HL gradient in a heart-shaped pattern. That'll correspond to MyoD expression, or muscle differentiation, in a heart-shaped pattern. And the remainder of the cells, which are too far from the HL gradient to be affected by it, will differentiate into neuron. So now I'll pass it over to Anand, who will talk about our approach to tissue patterning.
Thanks, Safi. So in order to actually pattern real cells onto real substrate media, there's 3 main steps. The first is to genetically engineer new communication and differentiation pathways in these cells. The second is to use a technology called laser direct write in conjunction with Craig Arnold's lab at Princeton to actually pattern these cells onto a substrate. And the third step is to use pattern formation and cell-cell communication to form these patterns. So as we can see here, in an example, there's 3 different regions of sender cells that have been patterned onto the substrate. And in the presence of receiver cells, we actually get this pattern. So this is an example that we've created using laser direct write with bacteria. And if you look in the upper right corner, you can see that the bacteria are actually living and growing. This is a single droplet on this pattern. In addition, we're using this technology to create patterns that are slightly more applicable for tissue engineering, as you can see here. And we're also using this to pattern mammalian cells. And in this example, we see a single mouse embryonic stem cell, and over a period of 4 days, you can see it growing to about 100 cells. So the second system I'm going to talk about now is this sender-receiver controlled toggle switch for cell fate. So in this case, in the presence of AHL sender cells, AHL will enter the system and cause the repression of TET-R, which pushes the system towards the lac-I state, which causes the expression of MyoD, the cell fate regulator for muscle precursor, as well as GFP as a reporter. Alternatively, when there's no senders in the system, IPTG and ATC can be used to control the toggle switch normally. These are some preliminary results from this system, where we have the CMV constitutive promoter expressing lac-I, and CAG-OP, which is a synthetic lac promoter expressing DS-RED. And as we see on the right, when there is IPTG in the system, we get expression of DS-RED, and when there's no IPTG in the system, we get low levels of expression. Here's another result from this system, where we have constitutive expression of TET-R, and we have a TET operator controlling the expression of GFP on the right. So as we see on the right with ATC, when there's high levels of ATC, we get GFP expression, and when there's no ATC, we get low levels of GFP. However, as you probably know, when you're trying to build the toggle switch, even this low level of GFP is somewhat worrisome, because you need high levels of repression for the toggle switch to function properly. So Saram in our lab fused the crab repression domain in these mammalian cells to the TET-R protein, which significantly improves this response, as you can see on the left. Here's another result from the same experiment, where you can see on the right that when there's no ATC in the system, the levels of fluorescence with TET-R crab are almost indistinguishable from background levels. The third system that we created and designed is a quorum sensing-based beta cell differentiation system to actually make cells that can produce insulin. And you can imagine a system like this will be pretty useful in the treatment of diabetes, where you'd be able to have cells automatically proliferate, grow, reach a critical density, and then automatically differentiate into insulin-producing beta cells. So the way the system works is that IPTG is added to the system to turn the system on, which causes the derepression of LuxI, which produces AHL. And this diffuses outside of the cell. And then AHL binds to LuxR, which activates GATA4 or SOC17. And these are cell fate regulators for the production of primitive endoderm tissue, which is the first step in producing beta cells. Here we're using the tissue-specific promoter AFP, which responds to the presence of alpha-fetoprotein, which is present in primitive endoderm tissue, and that controls the expression of DS-RED, which we use as a reporter. In addition, we have the expression of PDX1 and NGN3 under the control of PAFP, and that controls the expression of the formation. Those are cell fate regulators for the formation of pancreatic beta cells. And here we have another tissue-specific promoter, the mouse insulin promoter, which controls GFP to allow us to confirm that the cells have successfully transformed into insulin-producing beta cells. So as I mentioned, a system like this is pretty useful for the treatment of diabetes, where, for example, you'd actually be able to take these cells and transplant them into the pancreas and allow them to grow, reach a critical density, and then start to form insulin. So we've actually gotten a number of these components working so far in in vitro, where you have we've gotten the sender and receiver, as you saw earlier, as well as another component, which is the tissue-specific promoter component, where we have the AFP promoter expressing DS-red. And in these cells, which contain alpha-fetoprotein, you see we do indeed get this expression of DS-red. So to summarize, we verified that we have artificial cell-cell communication in mammalian cells. We designed 3 systems for regulating cell fate as a basis for future clinical applications: a communication-based multiplexer, a toggle switch to control cell fate, as well as a quorum sensing-based beta cell differentiation. And we've demonstrated that we have regulatory control of gene expression and differentiation, as well as we are able to form patterns using precise cell placement. So far, we've designed 25 biobricks and constructed them this summer, and these are going to be part of the stem cell biobrick registry. We'd like to acknowledge a number of people and groups that have helped us along the way, specifically and especially Sil, Christoph, Sairam, Ernesto, Professor Lemishka, and of course Professor Weiss. And this is a photo of our team from this summer. That's it.